The question number 5 is cell divides in a series of stages called the cell cycle. The stage 2 of the cell is called mitosis. The figure number 10 shows a simplified cell cycle for the human body cell. So a model is given here, the stage 1, when the cycle will start. The stage 2 is coming, the stage 3 is coming there. So this question 5.1 is that you need to draw one line from each stage in the cell cycle to what happening during the stage there. So we have different options right now. The students must have the knowledge of the mitosis and cell division. Three different stages are given in this model. If you look there, stage 1, stage 2 and stage 3. The stage in the cell cycle, what happens during the stage? Stage number 1 is here in the cell cycle when the DNA is going to replicate itself. So draw one line. So we have drew one line from each stage. Stage number 1, the DNA is going to make its copy. Mean every single chromosome is going to make a copy of itself. Stage number 2 is mitosis, where the nucleus will divide, and in stage number 3 the cell will divide fully into two. This question, part number 2, is an AO2 question, and again from mitosis and cell division. So if we look here, the mass of the DNA in the human body cell at the start of the cell is 6 picograms. So it is mitosis. So what will happen there? The mass of the DNA will be in each of the new cell produced by the cell division. So the DNA, we already studied this thing in the first part, the DNA will divide. When the DNA will divide, because it makes its same copy, therefore the size and the mass of the DNA will remain same. If initially it is 6 picogram, at the end of the division it will remain 6 picogram. It's not going to increase and it will not going to decrease. So the correct answer is 6 picogram of the new cell after the cell division. The next question is coming about the stem cells, which is an AO1 question. The student must have the knowledge about the stem cells. The stem cells are undifferentiated cells. Which statement about the cells is correct? So we will go all the options which are given here. We have to pick one of the box. Animal cells, stem cells are found in meristems. The meristems are basically related to plants. So we can't pick this option. So what we can do, we will cross all the irrelevant options there. Animal stem cells divide by meiosis. No, it is by mitosis. So this option is not correct. The two options are left. Meristem cells in plants can differentiate throughout the life of the plant. It is correct information. But we will see this next option. The meristem cells in the plant can only differentiate into one type of the cell, which is not the correct option, because they can differentiate into the roots, the shoots, or the leaves. Therefore, the growth of the plant is taking place throughout its life. So this is not the correct information. So the correct option is the meristem cells in the plants can differentiate throughout the life of the plant. Before 5.4, the lot of the information is given that is stem cells from the human embryos, okay, can differentiate into most type of the human cell. So human embryos are given here, and what is going on, they can differentiate into the human cells. Research is being done into the use of the embryonic stem cells in the medical treatment. The long-term effects of using embryonic stem cells in the patient are not well understood. In therapeutic cloning, human embryos are produced using a donated human egg cell and the cell from the patient. Now, this is the key information is given here. The embryo produced contain the same genetic information as the patient, which is a good thing. Stem cells are taken from the embryo and stimulated to divide to form the cells that the patient needs. The embryo is then destroyed. Now you have to keep this information when you're giving the answer. The stem cells are taken from the human embryos and we are using them for the treatment. At the end, we destroy an embryo. 5.4 suggests two advantages of therapeutic cloning. So this topic is coming from the stem cells, cell differentiation, and genetic engineering. The first advantage of therapeutic cloning is the therapeutic cloning helps in treatment of the diseases such as the Parkinson's diseases, spinal cord injuries, and diabetes by generating healthy cells and replacing the old damaged cells. The second advantage which therapeutic cloning can help us, that therapeutic cloning produces genetically identical cells to the patient cells. So they are less likely to be rejected by the patient immune system. We have here the therapeutic cloning helps in the treatment of the diseases such as Parkinson's diseases, spinal cord injuries, and diabetes by generating the healthy cells and replacing the old damaged cells. Second advantage can be the therapeutic cloning produces the genetically identical cells to the patient cells. Therefore, they are less likely to be rejected by the patient immune system. The last part of this question is 5.5 suggests two disadvantages of therapeutic cloning. We have to give the disadvantage of this. We see our model answer. First of all, if you look here, that is destroying an embryo. So therapeutic cloning involves the destruction of the human embryo to obtain the stem cell, which can raise a lot of the ethical concerns of many people. So therapeutic cloning involves destruction of the human embryos to obtain the stem cells, which raise the ethical concerns of many people. The second disadvantage is, you can write this thing in your answer, the process of therapeutic cloning is complex, technical, and which increases the chances of error and complications in the body. So in our model answering, the therapeutic cloning involves destruction of the human embryos to obtain the stem cells, which raises the ethical concerns of many people, because as a result of that, we are taking the life of an embryo. The process of therapeutic cloning is complex and technical, which increases the chances of the errors and the complications in the body.